Today I'm going to be talking about a process called protein synthesis. It starts with transcription and we start in the nucleus. The length of DNA that is going to be transcribed is called the transcription unit. To start the process, DNA unwinds. Then the RNA polymerase copies the DNA sequence. The polymerase copies the sequence towards the 5' end into mRNA. While doing this, the polymerase reads the nitrogenous bases and makes sure the RNA bases find their match. As all of this is happening, the RNA rezips the DNA behind it and allows the mRNA to move away. Eventually, the RNA reaches something called a termination signal that tells it to pull off. Before the mRNA can leave the nucleus, it has to have some refinement first. The first thing that happens is a special type of guanine is added to the 5' end. This is referred to as the 5' cap. On the 3' end, there is a poly-A tail added, which is made up of around 250 adenines. These caps protect the mRNA and make it easier to connect with other organelles. The final step before the mRNA can leave the nucleus is to cut out all of the extra information. This is called RNA splicing. The spliceosome allows the good stuff, exons, to be spliced together and the junk, introns, to be recycled. Now that all of these steps are completed, the mRNA leaves the nucleus. The second step in this process is translation. In this process, mRNA gets fed into the ribosome. Ribosomes are made up of proteins and rRNA. The rRNA is a binding site that lets the mRNA interact with tRNA. tRNA translates from the language of nucleotides to amino acids and proteins. On one end of tRNA is an amino acid, and on the other is a sequence of 3 nitrogenous bases known as an anticodon. Now that we've identified all the basics, we get to the actual process. During this process, the mRNA starts at the 5' end and goes through the ribosome. The ribosome reads the letters 3 at a time. These 3 letters are known as a triplet codon. The ribosome then finds the matching piece. This matching piece is a tRNA with 3 bases that pair with the triplet codon. These bases are known as the anticodon. The ribosome is also bringing in the amino acid on the tRNA when it does this as well. As the RNA goes to the ribosome, the amino acids all get together, starting a polypeptide chain. Once this process is complete, the amino acids go onto protein folding. Once the amino acids go onto protein folding, the process of protein synthesis is complete.